General physiology of kidney. Introduction. Kidneys are a pair of bean-shaped organs along the posterior wall of abdominal cavity. They are retroperitoneal organs which are located from the 11th thoracic vertebra to 3rd lumbar vertebra. The normal size of adult kidney is 11 to 15 cm. The left kidney is located slightly higher than the right kidney. Kidneys are surrounded by a layer of adipose that hold them in place and protect them from physical damage. Physiological anatomy of kidneys. General organization of kidneys and urinary tract. The two kidneys lie on the posterior wall of the abdomen, outside the peritoneal cavity. Each kidney of the adult human weighs about 150 grams and is about the size of a clenched fist. The medial side of each kidney contains an indented region called the hilum through which pass the renal artery, veins, lymphatics, nerve supply, and ureter. The kidney is surrounded by a tough fibrous capsule that protects its delicate inner structures. If the kidney is sectioned, two regions are seen: an outer part, called the cortex, and an inner part, called the medulla. The outer cortex contains many capillaries, while medulla consists of renal pyramids by renal columns. Pyramid contains minor calyces which unite to form a major calyx. Major calyces form renal pelvis, which function to collect urine and then transports urine to the ureters. Structures of nephron. Nephron is the functional unit of kidney. Each kidney in the human contains about 1 million nephrons, each capable of forming urine. Each nephron contains a tuft of glomerular capillaries called the glomerulus, through which a large amounts of fluid are filtered from the blood. A long tubule in which the filtered fluid is converted into urine on its way to the pelvis of the kidney, which consists of urinary tubules. They are proximal convoluted tubules, descending and ascending lobes of Henle, distal convoluted tubule, and collecting duct. The kidney cannot regenerate new nephrons. Therefore, eventually, there is a gradual decrease in nephron number. Renal malpighian corpuscle. Bowman's capsule surrounds the glomerulus. It is the location where glomerular filtration occurs. The filtration passes into the urinary space, then into the proximal convoluted tubule. Nephron tubules. Proximal convoluted tubule lies by single layer of cuboidal cells with millions of microvilli which function to increase surface area for reabsorption. The functions of proximal convoluted tubule are obligatory reabsorption and secretion. Loop of Henle. Fluid passes from proximal convoluted tubule to loop of Henle. The function of descending limb is water reabsorption, while the function of ascending limb is active transport of sodium ions, and it is impermeable to water. Distal convoluted tubule contains few microvilli. The functions of distal convoluted tubule are facultative reabsorption and secretion. It terminates in collecting duct. Collecting duct receive fluid from distal convoluted tubule of several nephrons which then passes through renal pyramid into minor calyx. Function of collecting duct is reabsorption of water. Types of nephron. There are two types of nephron, cortical nephron and juxtamedullary nephron. Cortical nephrons are about 85% of the nephrons. They have glomeruli located in the outer cortex. They have short loops of Henle that penetrate only a short distance into the medulla, and the entire system is surrounded by an extensive network of peritubular capillaries. Juxtamedullary nephrons are about 10 to 15% of the nephrons. They have glomeruli that lie deep in the renal cortex near the medulla and have long loops of Henle that dip deeply into the medulla. In some cases, all the way to the tips of the renal papillae. The long efferent arterioles extend from the glomeruli down into the outer medulla and then divide into specialized peritubular capillaries called vasa recta that extend downward into the medulla, lying side by side with the loops of Henle. Like the loops of Henle, the vasa recta return toward the cortex and empty into the cortical veins. This specialized network of capillaries in the medulla plays an essential role in the formation of concentrated urine. Renal blood supply and vasa recta. The blood flow to the two kidneys is normally about 22% of the cardiac output, or 1,200 milliliter per minute. The renal artery enters the kidney through the hilum and then branches progressively to form the interlobar arteries, arcuate arteries, interlobular arteries, also called radial arteries, and efferent arterioles, which lead to the glomerular capillaries. The distal ends of capillaries of each glomerulus coalesce to form the efferent arteriole, which leads to a second capillary network, the peritubular capillaries, that surrounds the renal tubules. The renal circulation is unique in that it has two capillary beds, the glomerular and peritubular capillaries, which are arranged in series and separated by the efferent arterioles, which help regulate the hydrostatic pressure in both sets of capillaries. By adjusting the resistance of the efferent and efferent arterioles, the kidneys can regulate the hydrostatic pressure in both the glomerular and the peritubular capillaries, thereby changing the rate of glomerular filtration, tubular reabsorption, or both in response to body homeostatic demands. The peritubular capillaries empty into the vessels of the venous system, which run parallel to the arteriolar vessel and progressively to form the interlobular vein, arcuate vein, interlobar vein, and renal vein, which leaves the kidney beside the renal artery and ureter. From the diagram, we can see that the blood flows from the aorta to renal artery, segmental artery, interlobar artery, arcuate artery, cortical radiate artery, efferent arterioles, then into the glomerulus. From glomerulus, the blood flow into the efferent arteriole, peritubular capillaries or vasa recta, cortical radiate vein, arcuate vein, interlobar vein, renal vein, and finally into the inferior vena cava.
Each kidney is supplied with blood by a single renal artery that arises on its respective side of the aorta before dividing into 5 segmental arteries that enter the hilus. Within the kidney, each segmental artery branches into several lobular arteries. The lobular arteries further subdivide to form interlobular arteries, which branch off into afferent arterioles. Blood flows into the glomeruli through the afferent arterioles. Blood flows out of the glomerulus through the efferent arteriole. The afferent and efferent arterioles regulate glomerular capillary pressure by selectively dilating or constricting. The kidney's venous blood, now filtered, flows from the glomerulus via the efferent arterioles into the peritubular capillary network, a low-pressure, reabsorptive system surrounding all portions of the tubules. This arrangement permits rapid movement of solutes and water between the fluid in the tubular lumen and the blood in the capillaries. The peritubular capillaries rejoin to form the venous channels by which blood leaves the kidneys and empties into the inferior vena cava.
Nervous supply of the kidneys. The kidneys are richly innervated by sympathetic nerve fibers which travel to the kidneys mainly in the 10, 11 and 12 thoracic spinal nerves and first lumbar spinal nerve. Stimulation of sympathetic fibers cause constriction of renal blood vessels and fall in renal blood flow. Sympathetic nerve fibers also innervate tubular cells and may cause an increase in sodium ions reabsorption by a direct action on these cells. Stimulation of sympathetic nerves increase the release of renin by the kidneys. The kidneys also innervated by parasympathetic nerve fibers which are the renal branches of vagus nerve. Functions of kidney. Kidney has the following functions: excretory function, homeostatic functions, endocrine functions, and endocrine metabolic function. For excretory function, kidney excretes drugs, toxins, and waste products. For homeostatic function, kidney maintains the water balance, electrolyte balance, acid-base balance, pH balance, volume of blood plasma, arterial pressure, and osmotic pressure. For endocrine function, kidney secretes prostaglandins. Renin is secreted by juxtal glomerular cell, while erythropoietin hormone is secreted by endothelial cells of peritubular capillaries of renal cortex. The same way for endocrine metabolic function. Kidney converts vitamin D3 to active 1,25-dihydroxycholecalciferol by alpha-1 hydroxylase enzyme in cells of proximal convoluted tubule under effect of parathyroid hormones. Steps of urine formation. First step, glomerular filtration. Second step, tubular reabsorption. Third step, tubular secretion. The glomerular filtration, the first step in urine formation. Glomerular capillary membrane. Glomerular capillary membrane has three major layers: endothelium of the capillary, basement membrane, and epithelial cells layer. For endothelium of the capillary, the pores are large fenestrae. It is 100 to 400 times more permeable to plasma, water, and dissolved solutes than capillaries of skeletal muscles. The pores are small enough to prevent red blood cells, platelets, and white blood cells from passing through the pores. The basement membrane is thin glycoprotein layer with negatively charged. The layer of epithelial cells or podocytes surrounding the outer surface of the capillary basement membrane consists of foot pedicels form small filtration slits. It is the passageway through which filter molecules must pass. Mechanisms of glomerular filtration. Glomerular filtration is a process of bulk flow driven by the hydrostatic pressure of the blood. Passage through the filtration membrane is limited not only on the basis of size but also by electrical properties. Small molecules pass rapidly through the filtration membrane, while large proteins and blood cells are kept out of the capsular space. This video shows the process of glomerular filtration.
Filtration is the first of three main processes in urine formation. Blood flowing from the glomerulus exerts pressure, and this glomerular blood pressure is high enough to push water and dissolved substances out of the glomerulus and into the Bowman's capsule. This fluid then becomes known as the glomerular filtrate. The wall of the glomerulus contains pores and is similar to a strainer. The diameter of the pores determines which substances flow through and which remain. The only blood constituents that do not move into Bowman's capsule are the larger molecules such as blood cells and most plasma proteins, which exit the efferent arteriole. To summarize filtration, glomerular blood pressure causes filtration through the glomerular capsular membrane to create the glomerular filtrate.
Determinants of glomerular filtration rate. Glomerular filtration rate is the quantity of glomerular filtrate formed in all nephrons of both kidneys per minute. The normal value in women is 115 milliliter per minute, while in men is 125 milliliter per minute. The glomerular filtration rate is determined by, first, the sum of the hydrostatic and colloid osmotic forces across the glomerular membrane, which gives the net filtration pressure. The glomerular capillary filtration coefficient. Expressed mathematically, the glomerular filtration rate equals to the products of glomerular capillary filtration coefficient and the net filtration pressure. The net filtration pressure represents the sum of the hydrostatic and colloid osmotic forces that either favor or oppose the filtration across the glomerular capillary. These forces include, first, hydrostatic pressure inside the glomerular capillaries, glomerular hydrostatic pressure, which promotes filtration. Second, hydrostatic pressure in Bowman's capsule, outside the capillaries, which oppose filtration. Third, the colloid osmotic pressure of the glomerular capillary plasma proteins, which opposes filtration. Fourth, the colloid osmotic pressure of the proteins in Bowman's capsule, which promotes filtration. Under normal conditions, the concentration of protein in the glomerular filtrate is so slow that the colloid osmotic pressure of the Bowman's capsule fluid is considered to be zero. The glomerular filtration rate can therefore be expressed as the following equation. Tubular reabsorption. For a substance to be reabsorbed, it must first be transported across the tubular epithelial membranes into the renal interstitial fluid and then through the peritubular capillary membrane back into the blood. There are three types of transport: transcellular route, paracellular route, and bulk flow. Transcellular route is where the water and solutes can be transported either through the cell membranes themselves. Paracellular route is the transport through the junctional spaces between the cells. Bulk flow is the transportation of water and solutes through the peritubular capillary walls into the blood after absorption across the tubular epithelial cells into the interstitial fluid. It is mediated by hydrostatic and colloid osmotic forces. Passive transport mechanism. Passive transport is transport of substances according to the concentration gradient and there is no need energy expenses. Osmotic flow of water occurs through the so-called tight junctions between the epithelial cells as well as through the cells themselves. This is especially true in the proximal tubules, which have a high permeability for water and a small but significant permeability to most ions such as sodium, chloride, potassium, calcium, and magnesium. As water moves across the tight junctions by osmosis, it can also carry with it some of the solutes, a process referred to as solvent drag. When sodium is reabsorbed through the tubular epithelial cell, negative ions such as chloride are transported along with sodium because of electrical potentials. That is, transport of positively charged sodium ions out of the lumen leaves the inside of the lumen negatively charged, compared with the interstitial fluid. This causes chloride ions to diffuse passively through the paracellular pathway. Urea is also passively reabsorbed from the tubule, but to a much lesser extent than chloride ions. As water is reabsorbed from the tubules by osmosis coupled to sodium reabsorption, urea concentration in the tubular lumen increases. This creates a concentration gradient favoring the reabsorption of urea. However, urea does not permeate the tubule as readily as water. In some parts of the nephron, especially the inner medullary collecting duct, passive urea reabsorption is facilitated by specific urea transporters. Active transport mechanism. Active transport is the transport of substances against the concentration gradient where requires energy in the form of ATP. Primary active transport. Primary active transport is the transport that is coupled directly to an energy source, such as the hydrolysis of adenosine triphosphate, ATP. A good example of this is the sodium-potassium ATPase pump that functions throughout most parts of the renal tubule. Secondary active transport. Secondary active transport. This often involves counter-transport of the substance with sodium ions. In counter-transport, the energy liberated from the downhill movement of one of the substances, for example sodium ions, enables uphill movement of a second substance in the opposite direction. One example of counter-transport is the active secretion of hydrogen ions coupled to sodium reabsorption in the luminal membrane of the proximal tubule. In this case, sodium entry into the cell is coupled with hydrogen extrusion from the cell by sodium-hydrogen counter-transport. This transport is mediated by specific protein in the brush border of luminal membrane as sodium is carried to the interior of the cell. Hydrogen ions are forced outward in the opposite direction into the tubular lumen. Pinocytosis. Pinocytosis is an active transport mechanism for reabsorption of proteins. In some parts of the tubule, especially the proximal tubule, reabsorb large molecules such as proteins by pinocytosis. In this process, the protein attaches to the brush border of the luminal membrane, and this portion of the membrane then invaginates to the interior of the cell until it is completely pinched off and a vesicle is formed containing the protein. Once inside the cell, the protein is digested into its constituent amino acids, which are reabsorbed through the basolateral membrane into the interstitial fluid. Because pinocytosis requires energy, it is considered a form of active transport. Transport maximum for substances that are actively reabsorbed and actively secreted. Transport maximum for substances that are actively reabsorbed. An example is the glucose transport system in the proximal tubule. Normally, measurable glucose does not appear in the urine because essentially all the filtered glucose is reabsorbed in the proximal tubule. However, when the filtered load exceeds the capability of the tubules to reabsorb glucose, urinary excretion of glucose does occur. In the adult human, the transport maximum for glucose averages about 375 mg per minute, whereas the filtered load of glucose is only about 125 mg per minute. With large increases in glomerular filtration rate and or plasma glucose concentration that increase the filtered load of glucose above 375 mg per minute, the excess glucose filtered is not reabsorbed and passes into the urine. Transport maximum for substances that are actively secreted. Substances that are actively secreted also exhibit transport maximums. For example, creatinine, 16 mg per minute. Para-aminohippuric acid, 80 mg per minute. Tubular transport maxima. It is the maximum amount of substance in milligram which can be reabsorbed or secreted per minute. Tubular transport maxima of glucose with normal blood glucose level equals to 375 mg per minute for men, and for women is 300 mg per minute. Reabsorption and secretion along different parts of nephron. Proximal tubular reabsorption. Total concentration of solute equals to 300 milliosmol per liter. Reabsorption of water by osmosis cannot occur without active transport. Concentration of sodium ion in glomerular ultrafiltration is equals to 300 milliosmol per liter. At proximal convoluted tubule, epithelial cells have lower sodium ion concentration. Due to low permeability of plasma membrane to sodium ion, active transport of sodium ions out of the cell by sodium-potassium ATPase pumps and favor sodium ion concentration gradient. Thus, sodium ions diffusion into the cell. Sodium-potassium ATPase pump located in basal and lateral sides of cell membrane creates gradient for diffusion of sodium ions across the apical membrane. Sodium-potassium ATPase pump extrudes sodium ion, creates potassium difference across the wall of the tubule with the lumen as negative pole. Electrical gradient causes chloride ions movement towards higher sodium ion concentration, and water follows by osmosis. Salt and water reabsorption in proximal tubule. From here, we can see that sodium ions is reabsorbed via active transport, while chloride ion is reabsorbed via passive transport, and water follows salt by osmosis. Counter current multiplier. In order for water to be reabsorbed, interstitial fluid must be hypertonic. Osmotic pressure of the interstitial fluid is 4 times than that of plasma. This results partially from the fact that the tubule bends permitting interaction between the descending and ascending limbs. Proximal tubular secretion. At proximal tubule, secretion of organic acids and bases such as bile salts, oxalate, urate, and catecholamines take place. Many of these substances are the end products of metabolism and must be rapidly removed from the body. The secretion of these substances into the proximal tubule plus filtration into the proximal tubule by the glomerular capillaries and the almost total lack of reabsorption by the tubules, all combined, contribute to rapid excretion in the urine. Para-aminohippuric acid also secreted by proximal tubule. Para-aminohippuric acid is secreted so rapidly that The average person can clear about 90% of the para-aminohippuric acid from the plasma flowing through the kidneys and excreted it in the urine. Kidneys secrete many potentially harmful drugs or toxins directly through the tubular cells into the tubules and rapidly clear these substances from the blood. In the case of certain drugs, such as penicillin and salicylates, the rapid clearance by the kidneys creates a problem in maintaining a therapeutically effective drug concentration. Solute and water transport in the ascending loop of Henle. Sodium chloride is actively extruded from the ascending limb into the surrounding interstitial fluid. Sodium ions diffuses into tubular cell with the secondary active transport of potassium ions and chloride ions. It occurs at a ratio of 1 sodium ion and 1 potassium ion to 2 chloride ions. Sodium ions actively transported across the basolateral membrane by sodium-potassium ATPase pump. Chloride ion passively follows sodium ion down electrical gradient. Potassium ion passively diffuses back into filtrate. The ascending walls are impermeable to water. Solute and water transport in the descending loop of Henle. At deeper regions of medulla, the osmolarity reaches 1400 milliosmol per liter. Descending loop of Henle is impermeable to passive diffusion of sodium chloride, but it is permeable to water. Hypertonic interstitial fluid causes water movement out of the descending limb via osmosis, and water enters capillaries. Fluid volume decreases in tubule, causing higher sodium concentration in the ascending limb. Countercurrent multiplier system. The countercurrent multiplier system multiplies the concentration of interstitial fluid and the concentration of descending limb fluid. It flows in opposite direction in the ascending and descending limbs. Close proximity of the two limbs allows interaction for positive feedback mechanism to take place. Reabsorption and secretion in the distal tubules. The very first of the distal tubule forms part of the juxtaglomerular complex that provides feedback control of the glomerular filtration rate and blood flow in the same nephron. The next part of the distal tubule is highly convoluted and has many of the same reabsorptive characteristics of the thick segment of the ascending limb of the loop of Henle. That is, it avidly reabsorbs most of the ions including sodium, potassium and chloride, but is virtually impermeable to water and urea. For this reason, it is referred to as the diluting segment because it also dilutes the tubular fluid. Approximately 5% of the filter load of sodium chloride is reabsorbed in the early distal tubule. The sodium chloride co-transport moves sodium chloride from the tubular lumen into the cell, and the sodium-potassium ATPase pump transports sodium out of the cell across the basolateral membrane. Ions diffuses out of the cell into the renal interstitial fluid through chloride channels in the basolateral membrane. The thiazide diuretics, which are widely used to treat disorders such as hypertension and heart failure, inhibit sodium chloride co-transport. Late distal tubule and cortical collecting tubule. The second half of the distal tubule and the subsequent cortical collecting tubule have similar functional characteristics. Anatomically, they are composed of two distinct cell types, the principal cells and the intercalated cells. The principal cells reabsorb sodium and water from the lumen and secrete potassium ions into the lumen. The intercalated cells reabsorb potassium ions and secrete hydrogen ions into the tubular lumen. Secretion. Organic acids such as phenolate, para-aminohippuric acid, and bases such as choline are secreted at the proximal convoluted tubule, while potassium ions are secreted in the terminal parts of the distal and collecting tubule. Secretion of substances from the peritubular capillaries into the interstitial fluid, then transported into the lumen of tubule and into the urine.